So let's see how the loose connective tissue looks like in the macroscope, along with the other dense connective tissue, the two forms, the dense regular and dense irregular. For this example, we're going to use the skin, which is an ideal section to see all three types of connective tissue. So first up, we have just a short description of where we're going to find the loose connective tissue and where we're going to find the dense connective tissue. So in the epidermis, we'll discuss this epidermis, and we already mentioned some parts of the epidermis in the epithelial video. So now let's focus on the epidermis, the pure layer of true skin. So first up, we have the papillary layer, and the term papillary layer is named as such because of the presence of these dermal papillae, these projections that are that project over the epidermis. And this is because in this manner, whenever we have a section, any part of tissue that projects onto another, this is to serve typically the point of to increase surface area. So in some cases, we're going to say that this increase of surface area serves diffusion reasons or serves purposes of other sorts. In this case, we have the point of this the existence of this papilla is to increase the attachment of the epidermis on the skin. For example, if you imagine that this papilla, these projections were not present in this area, we just simply have a straight flat area. The surface area, let's say for example, it's 5 centimeters. But the moment that this straight line becomes a wave-like appearance, typically called papilla, this papilla increases the surface area to actually multiple times of that of 5 centimeters than just the 5 square centimeters that would be only if it was the case of a flat presence. So this is the purpose of the papilla. And in the papillary layer, we're going to find the loose connective tissue. We'll discuss in a second. We'll see actually how it looks in a microscope in just a second. So at this point, beneath the papillary layer, we're going to find the reticular layer. We already discussed the term reticular, and it means network. And this is because the reticular layer of the skin is filled with many glands, sweat glands, many eccrine glands, and so on, along with other sebaceous glands and things we will discuss specifically in the skin video. So at this point, the reticular layer is filled with dense connective tissue, along with vessels, nerves, and many other structures. At this point, we're just going to focus on the dense, irregular connective tissue of the reticular layer. And lastly, in the subcutaneous layer, the lower layer, the hypodermis also called, we can find typical presence of loose connective tissue along with adipose tissue. We're going to see the adipose cells, specifically the white adipose cells, right in the side, right in the slides right now. So let's start first up with the skin. This is the typical H&E section of the skin, and in this case we can find the papillary layer right here, exactly beneath the stratum basale, exactly beneath the layer of the epidermis. So let's zoom in. So here we're going to find the typical loose connective tissue. This is filled with, as we can see, small vessels, many fibroblasts, and a lot of collagen type 1 along with other collagen fibers. So in this case, in this area, we simply see the loose density of the connective tissue. Don't be confused. In all cases, the term loose and dense simply refer to the density of collagen fibers in the area that we're examining. So the distinction is not necessarily always 100% clear. We'll find areas that are more loosely dense, and some more looser densities, and some areas of way higher density. So it's not necessarily 100% accurate to term one connective tissue loose as dense. We simply term it in the tendencies of the density, not the precise densities. This is a pedagogic, this is an educational classification, not so much as a functional one. So exactly beneath this is, again, this is the area of the loose connective tissue, and exactly beneath we can find the dense connective tissue. This is the reticular layer. So in the reticular layer, we're going to find, again, the same cells, fibroblasts again, fibrocytes, and the same thing. But the difference is that in this case, the density and the amount and the mass of the collagen is way higher than that of connective tissue. Besides this difference, there is no other specific functional difference or, let's say, functional difference of contents. Nonetheless, because of the higher amount of collagen in the dense irregular connective tissue, this tissue is more resistant to stress. Stress simply means that application of forces in many different directions. In comparison, we're going to find the dense regular connective tissue. This is going to be in just a second. We're going to discuss the functional difference in the irregular and regular, but they both have to do with mechanical resistance. And again, in the case of the dense irregular connective tissue, we have increased resistance to stress. So we can see both of these tissues right here. And if we zoom in, for example, we can easily distinguish the fibroblasts and the fibrocytes. Typical example is right here, this one, this cell. This has the appearance of a fibrocyte, thinner again in comparison to the fibroblast, which is a bit thicker and wider, and also has excessive thicker amount of cytoplasm. So if we zoom out and we focus on the hypodermis, we're going to find the very big masses of adipose tissue. All of these are typical adipocytes, white adipose tissues, interspersed within the loose connective tissue, also called areolar in this case. So let's zoom in and actually discuss the morphology of the white adipose cell. So I'm looking for a find a better area. There we go. So in this zoomed area, we can find these vacuoles, these empty spaces right here. And of course, these empty spaces were the one big vacuole of lipid was present before the preparation of the slide tissue preparation, as we discussed already in a past video. And we can typically see the nuclei being punched out on the side in the periphery. For example, we can see a nucleus of adipose right here or right here. This is a much better location to see it. So there it is. Now, of course, we can see that this is empty place, this empty spaces again where the lipids used to be before the preparation of the slide. The morphology is very, very characteristic, and it's not very easy to confuse the adipose tissue. So let's check out the tendon in this case, the dense irregular connective tissue. Now, this is a section of the tendon. This is misnamed right here. So again, this is the dense irregular connective tissue, and we can see if we zoom in, again, the same contents, again, fibroblasts and fibrocytes, typical presence right here of a fibroblast and a fibrocyte in this case. And the differences, the major differences, is the parallelity of the collagen fibers. Now, in this case, we can see that the fibers are arranged in a parallel manner, and because of this arrangement, this tissue is more resistant to strain, which is flexion. Of course, this is exactly what we need in areas of tendons, again, because they transmit the force from the muscle to the bone, and this is exactly the ideal structure and morphology for this cell to actually, for these tissues to work properly and to function properly. And again, the irregular connective tissue, dense irregular, is resistant to stress, and the regular is resistant to strain, to pulling motions. So let's move on. Again, we talked about the tendon. Now let's talk a bit about the elastic tissue. We discussed the structure of the elastic fibers, how they function, and the typical example of the elastic tissue, in this case elastic fibers, is in the aorta. The aorta, of course, this is a very, very big vessel, in fact one of the biggest vessels of the human body. And in this area we can find the three layers, tunica intima, tunica media, and tunica adventitia. We'll see them right now in a second, and discuss just a bit in order to point out the unique portions of the connective tissue in this case. So this is a very nice section of the aorta, and we can find again the tunica intima on the top. Tunica media is the thickest layer of the aorta right here, and tunica adventitia is the last one on the outermost layer of the section. So in this case, we wanted to focus on the elastic tissue, so we're going to zoom in the tunica media, exactly in the middle. So the tunica media is nothing more than just layers and layers of elastic fibers, and then again smooth muscles. Elastic, smooth, elastic, smooth. Lasagna, if you prefer the term, lasagna layered structured, which is again layers of cells, fibers, cell, fiber. So again, the structure as we see it right here is again interspersed with the lamella, also called elastic lamellae, because of the presence of the repeated structure in the specific sequence again and again, and repeated sequence over and over again. And here we're going to find the elastic fibers along with the smooth muscles. The thing is that the unique feature is that, in fact, instead of the fibroblasts, here the cells that produce the fiber, the elastic fiber, is the smooth muscle cell. The smooth muscle cell typically has a different function, has a contractile function, not that of production. But in this case, in the aorta, we can see that these smooth muscles also produce these elastic fibers. And the function, the purpose of this is to allow the diameter to go back to its original size, because of course we have the blood coming out of the heart with high pressure, and we, of course, whenever we have a volume pressing through an area, there's a tendency to distend the vessel and the artery. So in order to maintain the diameter straight and similar, and to maintain the diameter constant somehow a bit, not of course fully constant, but there is some sort of pulsatile motion of the vessel. This retrieval back to the original size or diameter is a common function between the contraction of the smooth muscle cells and the elastic fiber in between these two layers. So in this section, we're going to find also the Verhoef staining, which allows us to visualize the presence of the elastic fiber, and the positivity, of course, is in this black color in between the layers. So if we zoom in, we can actually see clearly the amount of the elastic fibers interspersed between the smooth muscles. And again, in comparison, this is the H&E staining, and we can actually see the many numerous layers of smooth muscle cells, approximately 5 to 50 in the aorta, depending on the age and depending on many other factors. So typically there are up to 50 layers of, again, elastic lamellae and smooth muscles. This is the typical morphology of the smooth muscle. We can find the cigar-shaped nucleus. This is a typical morphology of the cell, and this allows us to actually visualize the cells much easier and to distinguish them between fibroblasts and smooth muscle cells, again based on the nucleus. This is again the same section. So we can move on to the reticular tissue. The reticular tissue, as I said before, again the term reticular stands for the network, in this way that it actually forms a network. And this is typically found in areas of immune systems. For example, the ideal example to visualize these reticular fibers along with the fibroblasts that produce them. Again, reticular fibers are collagen type 3. Specifically, they're agurophilic. They have an affinity to silver. And the cells that produce them are reticular cells, again, which are specialized fibroblasts. So we're going to find these sections inside the lymph node, and we should be able to distinguish the lymphocytes and the fibroblasts. So typically, this is the morphology of the lymph node, again, the cortex and the medulla. So we're going to focus somewhere in the middle, in between the medulla and the cortex, the paracortex also called. So in this area, we can zoom in a lot and we can visualize the two different populations of cells. The lymphocytes are typically identified because of their spherical nucleus and spherical shape and few amounts of cytoplasm within them. Surrounding them, we can actually see cells that have more typical morphology of fibroblasts. For example, this one is a fibroblast. Actually, here we're going to call it reticular cell. In fact, there is a silver impregnation method here. We can see the staining, and within the whole site, we can actually visualize all of the reticular fibers and the presence of reticular tissue. Of course, the point of this reticular tissue is to actually allow for the communication of the cells in between them, along with the antigen-presenting cells and the lymphocytes. This is the focus. This is the point of the presence of the reticular tissues and reticular fibers within them. So we can clearly see this black stains right here, the positivity along with the reticular fibers, and this is again how we can actually see the amount of reticular fibers interspersed and placed within the whole lymph node. Again, this is the typical morphology. So we can move on. This is again the typical morphology of the reticular cells and reticular fibers. This is again the staining, similar staining, where you visualize this. And that's it for today. Thank you very much.